Thanks. And our third panellist this morning is Professor John Hammond. John is the Director of Research and leads the Immunogenetics Group at the Pirbright Institute in the UK. He's also the Director of the Livestock Antibody Hub. His research focuses on how genetic variations shape immune responses in livestock to ultimately improve disease resistance and vaccine responses. He has a PhD in comparative immunology from the University of St Andrews and previously conducted research at Stanford University. John.
Great. Genuinely excited to be here because you're not my normal crowd. I'm immunology, I'm vaccinology, and so I don't have to convince you about why we're doing this. But my background is in infectious disease, and there is a lot of lessons that we can learn from how we've tried to apply high-resolution immunology to really intractable problems in infectious diseases. In the last 5 years and in the coming decade, a lot of new vaccines or a lot of improved vaccines will be coming to market because of the high-resolution capability of the methods we've been able to apply. There have been some challenging issues. There are still pathogens where there is no vaccine or other intervention available. So our role in this is a strategically funded institute in the UK. We're not commercially driven. We're very much knowledge-based. And so what we're trying to do is bring that knowledge of our infectious disease vaccinology research programs into the potential for a vaccine for methane reduction. And I guess I can't really see the screen very well from here, but the key parts I wanted to talk about are the antigen, the formulation, and the timing, because these are going to be critical in the success of any vaccine. So I thought it would be useful to just be on the same page about how a vaccine should work. So, vaccine administration, many different routes. It doesn't really matter for this talk. It can come many ways. It can be an injection, muscular injection. You can inhale them. They can be implanted. They can be slow release. It doesn't really matter at the moment. But the vaccines that we've generally been developing have been against pathogens. So that vaccine contains a part of that pathogen, an inactivated pathogen, a weakened pathogen, something that the animal will recognize so that when it's actually infected, sees that pathogen, there's already a response. And importantly, part of that response is a memory response. There are antibodies that have been produced that are specific to that pathogen. They're laid down in the bone marrow or somewhere else, and so that animal can respond. However, there's some key challenges with what we're proposing to do with the methane vaccine. The first one is a little bit obvious, but methanogens aren't pathogens. They're already in the animal. There are actually antibodies already there that are probably developed so the animal can tolerate having methanogens and other microbiome microorganisms in the first place. So what we need to do is we need to challenge the animal in a way that doesn't actually break its own tolerance. We still need to have that memory response. Those antibodies still have to be specific, but they have to act in a component within the rumen with a mechanism that is unusual. Systemic, lung, mucosal surfaces, antibodies work in these places very well. They don't necessarily work in the rumen. And that's quite critical because the vast majority of antibody functions rely on other cell types, and those cell types aren't in the rumen. So really the mechanism of this vaccine that we're talking about has to be the physical interaction of those antibodies with those methanogens in a very specific way, which is a pretty unique challenge, I think. Not only that, is that there are many different flavors of antibody. In cattle, there are several different flavors of antibody. These bind to pathogens or whatever antigen we're talking about in different ways. They also have different functions. They can bind more strongly, they can bind more weakly, and they can induce different functions. So not only does our vaccine have to produce these antibodies, but it's got to produce the right antibodies that function in that compartment in the right way. And this is where the advances really come in over the last few years. So what we're able to do now is we're able to To immunize animals with antigens or methanogens, we essentially treat it like a pathogen, but rather than look and see if that's protective, what we do is we take those antibodies back out of the animal. So we take those antibodies from the blood, we can take them from the spleen, we can take them from the salivary glands. And when we isolate each individual antibody, what we do with those is we then sequence those antibodies. So we have those antibodies forever as genetic material. And the key part to that is that we can then make those antibodies again. But not only can we make them, we can engineer them. So we can actually turn one antibody from a cattle into any flavor of antibody we want. In fact, as Paul says, these antibodies, or the outcome of the vaccine-driven antibodies, need to work across multiple species. We can take an antibody from a cow, and we can make that into an antibody that looks like it's from a goat, from a human, anything you want, really. This engineering capability also allows us to manipulate how strongly those antibodies actually bind the methanogens in the first place, depending on what sort of antibody that is. So with our colleagues in New Zealand, what we're doing is that we're isolating and making these antibodies, and we're then assessing how well they function in terms of reducing methanogen growth or emissions from those methanogens. And this is an iterative process that leads us into a real antigen discovery structural guided approach, which tells us exactly what a vaccine needs to be able to do to have efficacy in this space. So really what we're attempting to do is to really design a proof of concept outcome of a vaccine, which we can then use to improve vaccines that Colin, Lucid and Bio are generating. We could take a vaccine that's 25% efficacious, and by understanding the exact antigen landscape and the type of antibodies that need to be induced, we could move that beyond the 30%. It's that high resolution detailed knowledge that's essential, I think, in such a complex system as this. Timing will also be key. So there are two elements to this, and I think how any vaccine will be deployed. So the first is early vaccination in calves may permanently reduce the frequency or the methanogen frequency in the microbiome, or that microbiome that may well persist through that animal's life, or through repeated vaccination may persist through that animal's life. That could have quite profound changes in how that vaccine is deployed and when you need to administer that vaccine in the first place as the microbiome is colonized, depending on what the dam's microbiome may be like. And the second component of this, of course, is that if you fundamentally alter the dam, then the calf's microbiome may well be altered for the rest of its life. And I think this will be my last slide, but the overlap with the genetics and the genetic selection in this space, I think, is critical, because there are significant epigenetic changes that I think could be driven through this approach. And over generations, I think it could be much more longer lasting than it would with just a vaccination program alone. Thank you.